biology, and welcome to a quick video going over the homework, which was the mitosis protein roles. So we talked about these 4 different steps yesterday or today in class, but I'm just going to reiterate the different phases and what we're looking at. So if we think about mitosis, one of the most important steps during interphase or non-cellular division is S phase. And S phase is where we copy all of the DNA in a double-sided DNA strand. So right over here is the parental DNA. This is what it looks like before synthesis. We unwind it using an enzyme called helicase. We separate it, and then we duplicate each side to make 2 copies of the DNA because each cell needs to have the same exact duplicated DNA. If they don't receive duplicated DNA, we're going to have issues in terms of mutations or tissue problems, etc., etc. Note that this strand is really the bottom strand, and we are making a new top strand. And alternatively, up here we have the top strand, which was complementary to this strand, and we're making a new bottom strand. So in a way, this is called the semi-conservative model, because one half of the strand is used to make the other half, and they are basically mirror images. So old, new, new, old. Old is top and bottom. New is top and bottom. So we're going to be able to create 2 strands of DNA using the enzyme DNA polymerase. So we're going to have 2 versions that are half and half new, half of the initial parental DNA, hence why it's called the semi-conservative model. Now, reminder that DNA has nitrogenous bases in the middle with the 5-carbon sugar in the phosphate group, and A's and T's and G's and C's are the base pairs that make up different sets of nucleotides, which gives us information. So I believe on the front page, your bottom part of the first page should have this complementary strand. I have not drawn the appropriate hydrogen bonds in between each, but A's and T's always go together and C's and G's always go together. So between these letters, you should make hydrogen bonds. This is what we're making over here. Top is the parental strand. Here's the daughter strand. The top is the daughter strand. The bottom is the parent strand. So on the backside, you should be drawing something that looks like this on the top of page 2, where you have 2 copies of DNA that look the same, but what is old versus what is new is different between the 2 strands, which is color-coded here. And now when it comes to helicase, helicase is going to be breaking apart these hydrogen bonds, which hold the 2 strands together. It does that with physical displacement, but it also uses chemical charges to interrupt the hydrogen bonds. So given that most of the hydrogen bonds have a very powerful negative charge with a weaker positive charge, you would want to think by looking at that image on your figure whether or not negative or positive charges in this enzyme would be more appropriate for disrupting these hydrogen bonds that keep the parent strand normally together. So the second protein and cycle that we talked about is G2. G2 is where we're getting prepared for division, and this is where we take our euchromatin and condense it down to heterochromatin. And we need to have 6 feet of DNA be compact into 46 condensed and separate chromosomes, much like you take your entire house and put them into boxes: kitchen box, bedroom box, toys box, video games box, etc., etc. Now we use histones to do this in terms of the condensing and wrapping up. And we use an 8 protein structure called the histone octamer, and then the H1 histone locks the DNA into place. Now, really important for understanding your questions about the histone is the structure of a nucleotide. The outer part of the DNA, which is the sugar phosphate backbone, sorry about that. Sugar phosphate backbone, here's my phosphate, here's my sugar, is what chiefly interacts with the environment, i.e. temperature or pH or other proteins. So you're going to want to think about what charges in these proteins would allow this DNA structure to want to interact so readily and bind so tightly to these proteins. Metaphase is the next place where we see a protein that's really important, and that's where the kinetochore proteins and the microtubule proteins that are coming from our centrioles attach at the centromere, and that again creates the tensile tug and pull of the metaphase lining up. And without these microtubules and kinetochores attaching, we would not be able to split apart duplicated chromosomes, i.e., we wouldn't be able to have mitosis occur. An old idea that we learned about back in Unit 2 is that two proteins during cell reception can interact based off of hydrogen bonds and mirror images of the charges. So that might be helpful for thinking about what is going on in these amino acids here and here that allow them to interact so tightly and so strongly in order to pull apart chromosomes that are pretty tied together using the proteins we'll talk about on the last image. This gets us to anaphase, which is our last step where proteins are really important. The enzyme separase is really important for breaking apart these cohesin proteins. And the cohesin proteins are found in the center of the centromere, and they tie together the duplicated copies of the chromosomes. So this chromosome and this chromosome are exactly the same information. They're duplicated copies. Separase, if we're looking at our flowchart of cell signaling, is normally inhibited from being freed and doing its job. So at the beginning, separase exists in this partnership with securin, which is another protein. Basically, it's locked up. It's not able to do its job. The cell makes these SACs, which are inhibitory proteins. We can call them SACs for short. And they are made when the kinetochores are not attached to microtubules. So this right here is an inhibition. As soon as kinetochores attach to all of the microtubules, so all of the chromosomes are attached, this step stops. This allows a different protein, CDC20, to rush in and do what's called ubiquitination. Ubiquitination. You do not need to worry about pronouncing that word. But basically, they add a bunch of small molecules to securin, denaturing it, which allows separase to become active and free. Separase being separated from securin will allow it to be free and be able to do the job of separating the cohesin proteins. And once the cohesin proteins are separated, we can reel in the two chromosomes and separate them into our daughter cells. Now, what do you need to know about this? Well, cohesin is an enzymatic protein. Separase, is an enzymatic protein, meaning that it has a binding and catalytic site that allows it to bind to a part of another molecule and do both physical and chemical reactions to break apart or react with another protein. So again, the idea of enzymes doing work by breaking things down using really positively and negatively charged proteins is really important for us understanding why these separase proteins can be broken apart. And going back to cell signaling, again, it's a flow chart of different steps that happen in the cell. First, we need to have microtubules attached to kinetochores. Then the inhibitory proteins are stopped. This allows separase to be freed, and then separase is able to go and break apart the sister chromatids. I hope this video has been helpful. We'll go over the questions in class tomorrow, and I hope you have a great day.